3 hours, 15 minutes of presentations of a seminar. I'm very happy because you are still there. I mean, more than 200 connected people at any given time, and thank you for your patience. Thank you, of course, to all the 20 participants. It's not, there's going to be one winner of the beautiful science and of the Game Changer Award, but I've seen dozens of beautiful science projects and dozens of Game Changer Awards. But we still have some things to comment before announcing the winners. And it is the result of the vaccine voting. So the audience has spoken, and I think that the winner is clearly the Oxford AstraZeneca vaccine. And I'm very happy because this is the opinion of around, I mean, we have like 100 votes for the vaccine that's going to be first approved by the EMA. And we have invited Dr. José Luis Cabero, and with a long experience in vaccine development. And hello, José Luis, good afternoon, good evening.
Good afternoon.
Thank you for joining us today. And this is like after the Barca matches, this is like the after match conversation. Mm-hmm. So how was the game? I mean, the match was not, I mean, there's a clear winner, and I would like to know, as an expert, your take on your opinion on the Oxford vaccines, on the difficulties of running this kind of trials, and what would be, if you can travel in time 1-2 years from now, how do you see that the population, the world population will be get vaccinated? Is going to be just one winner? Is it going to be a combination? Maybe, perhaps, just saying a combination of a vaccine with antibodies. So what's your take on this result and on the vaccine scenario right now?
First, Jordi, thank you for inviting me to contribute and to be part of this. I enjoy very much the presentations. I think it's a very good indication of the vibrance that is going around in relation to having ways to tackle this new pandemic that we are living with. Regarding the winner today, of course, it's not a surprise that the AstraZeneca Oxford vaccine is getting a lot of attention and adepts. It was one of the first runners actually to start clinical trials, and the team behind is very experienced, particularly in the chimpanzee adenovector that they are developing, which has the advantage of not having pre-existing immunity in humans as the other adenoviruses that are being used by other vaccines, like adenovirus 5, used by the CanSino company in China, and by the Sputnik vaccine in Russia, or the adenovirus 26 used by Johnson & Johnson, although the last one has much lower pre-existing immunity in humans, which is a much more rare adenovirus. It's very interesting that we are betting to a technology that is new, has been extensively used in clinical trials so far, and is being used in other clinical trials, but has not been reached the market yet. In spite of that, what we have seen regarding efficacy and safety is very promising. We have seen some pauses in some of the trials with the AstraZeneca Oxford vaccine that has been considered not to prevent further development. And in fact, the trials in the UK, in Brazil, in South Africa, and in India with that vaccine are ongoing, and in some geographies probably close to completion. Still is a pause in the US, and there are unofficial information or data going around indicating that they may resume the trials in the US next week. So all in all, these vaccines seems to be in the front runner phase, and it's likely that they will get approval in the EMA, one of the first. I'm not sure that it will be the first because, as everyone knows, the second runner-up, which is Moderna, and Pfizer BioNTech, are very, very closely approaching the finishing line, at least for an emergency use authorization. Now, Moderna just announced today that they have concluded the recruitment in their large study, and Pfizer and BioNTech a week ago announced that they started their rolling submission at the EMA in Europe. So it's a tight race between these three ones, with some advantages regarding the distribution in favor of the AstraZeneca Oxford vaccine, because the RNA-based vaccines will need a cold chain that to be distributed, that will require -80 degrees capabilities all the way, which definitely will put some logistical challenges to those of them.
Yeah, I see that we are starting to finalize the counting. Game Changer Awards, but before announcing them, and because this conversation with José Luis is very interesting, I would like to know, José Luis, what your opinion on the classical vaccines. Because, I mean, it seems like in this case, that would be the first option.
It's it's a very interesting question, because I said that it's very interesting to notice that we are betting on novel technologies, while one would like to believe that the more established traditional technologies like inactivated vaccines or recombinant subunit vaccines like the one from Novavax will have further chances of succeeding. And perhaps at the end, they will become more established. They will not be in the front runner. We have 9 vaccines in phase 3 clinical trials, but we have 42 in clinical trials as far as I'm aware of. And that means that we need to be taking a look at the vaccines that are in phase 2 today, among them one from Sanofi, and using an adjuvant from GSK, a proven adjuvant, that this is a BioNet vaccine as well. So I think still we will see the traditional vaccines probably acquiring an important role, not in the first phase, but an important role. The first phase will probably allow us to have vaccines for at-risk populations, and most likely to ameliorate the course of the disease, rather than to sterilize and to prevent the transmission of the virus, which will be needed at the end. So still is a way to go with vaccines. Monoclonal antibodies, as you said, will play a role at the very beginning together with the vaccines, but we will see first and second generation vaccines for a while, until probably 2 or 3 will become the ones standing out and becoming widely adopted.
Excellent. We may see in the future like combination, for example, starting with an inactivated vaccine and then giving a boost with an RNA, or the other way around. Absolutely. Maybe in the long term, this is probably a combination. And last question, José Luis, thank you for your insights. And as I promised to the audience, the aftermatch with you commenting on the results has a lot of value. What about manufacturing and reaching the population? One thing is approval, which we all hope is going to happen in the next few months. Next thing is the vaccines to be available for the general population at a global level. So.
Correct. I think this is very interesting, and again, is a good proof of what can be done when there is a will and urgency and a sense of urgency. So what we have seen in vaccine development is unprecedented. So the sequence of the SARS-CoV-2 virus was made available the 11th of January 2020. 10 months later, we have what we already have, an enormous amount of people working into different approaches to tackle the pandemic. And then particularly vaccines, we have never seen this. The fastest vaccine that we have seen in history is the mumps vaccine that took 4 years to be developed. Never seen these huge efforts. And among that is to increase manufacturing capacity at risk. So it's a lot of investment coming from different governments and non-profit foundations or organizations, some foundations like the Bill Gates Foundations, and a consortia of public and private partners trying to increase the manufacturing capacity. So we do see possibilities to tackle, and actually we expect to have a significant number of doses early next year and during the course of next year to target risk populations. But we need to manage expectations. The vaccine will not be the magical bullet to manage alone the pandemic in the near and medium term.
Yeah, and final comment. I mean, from your side that are actually working in the vaccine field, and also from my knowledge, and I think you would agree with this. I don't think we are compromising the safety of the study participants with the speed. It's not a speed at the expense of safety. I think that all the trials are keeping very strict pharmacovigilance and safety measures to protect the study participants. And really, it has a lot of merit to be able to deploy these complex clinical trials and at the same time keeping everybody safe and really studying all the potential adverse events connected with the eventual use of the vaccine. We have seen several trials being paused several times just because one case among the 15,000 showed some kind of safety signal, right?
Yeah, I think it's a very, very legitimate comment, and I think we have seen that all the sponsors are taking the care that is needed to watch safety. But we need to remember that probably the first authorizations will be for emergency use, and the trials will continue in order to increase the safety information. Still, one thing that will be needed to be looked after is the cases that are very rare, but they do exist or they have occur in some vaccines, which is the vaccine-dependent disease enhancements or antibody-dependent enhancement, which is the fact that some pre-existing immunity or the production of non-neutralizing antibodies may contribute to enhance the disease. And in order to see this, we will need to see much larger exposure to individuals. But this is a matter of time and exposure, not a matter of neglecting this at this point.
Excellent, Jose Luis. Thank you very much for your time. Thank you very much for your wise comments. So the moment of truth has arrived.
Thank you.
Are you prepared? Are you prepared to know the winners of the? First of all, we'll announce the winner of the Beautiful Science Award of the Chasing COVID webinar, which goes to Nuria Montserrat from IBEC for her project, Engineering Mini Organs to Understand and Target SARS-CoV-2 Infection. So Nuria, congratulations, and thank you very much for your contribution.
Thank you. Thank you very much for the organization of this wonderful meeting, for letting us space for explaining the science that we do all together. And this is not the merit of only part of my institution. So I'm very thankful for this moment. Thank you. Thank you very much. And for all the attendees and to everybody. This is really nice. Thank you.
Okay, I don't know, we have kind of an animation to celebrate the prize. The Girona Houston control did.
With this Zoom, I don't know. I think that some people can even put some stuff back.
Okay, so thank you, Nuria. I think it's a beautiful project. It's a beautiful science. It's really creative, innovative, and not only for SARS-CoV-2, but for many other diseases and viral diseases. I mean, this concept of organoids as models, quick models, where to test vaccines, developments, antibodies, etc., is really very it's going to bear many fruits in the future. Thank you very much. And the next and the final prize award for the game changer. I think we are all in agreement that besides vaccines, what we need right now are quick, affordable, reliable tests, diagnostic tests. So the winner of the game changer is, Maria Soler from ICN2 for his project Convat, that was, in particular, was featured in one of the latest issues of Nature Biotechnology as one of the game changers, diagnostic tests that can change the course of the disease in the following months. So congratulations, Maria. Congratulations to your team.
Yeah, thank you. And yeah, that's it. It's a big team, and we are working very hard for this. So yeah, thank you very much. Thank you for the opportunity for sharing the research with such amount of people. And yeah, we are open for collaboration, for investment, for anything. So if you just want to know more about the project, just text, send an email, phone call, whatever, and we'll be here. So, and yeah, thank you very much.
So thank you, everybody, and thank you for your patience, and thank you for being here. I think that we have seen excellent science, beautiful science, game changers, and what makes me particularly happy is a lot of interaction between the scientists and all the participants in this webinar. So, as I promised, it was long, but I hope it was worth it. So, goodbye, everybody. Take care, stay safe, and see you soon.